Good morning, one and all.
Good morning,
Good
sir.
morning, sir.
Myself,
Good
Vai
morning,
Kumar.
sir.
Myself, Vai Kumar, Assistant Professor on contract basis in Department of Botany, Dr. B.R. Ambedkar Open University. Honorable Vice Chancellor of Dr. B.R. Ambedkar Open University, Professor Sitaram Rao sir, and dedicated registrar, Dr. Lakshma Reddy sir, and revered academic director, Professor Sudharani Madam, and respected Dean Faculty of Science and in charge at the Department of Botany, Professor Pushpa Chakrapani Madam, and today's chief guest and resource person, Honorable Professor Manohar Chari sir, and all respected director and deans and faculty member and counselor teachers and dear students, a warm welcome you all to this webinar. I'm very happy to share with you the Department of Botany has initiated webinar series. And this is the first webinar organized by Department of Botany and Faculty of Science. So I'm very grateful to Honorable Professor Manohar Chari sir for accepting give a lecture on biology to biotechnology. Biology to biotechnology. Now I request our Dean Faculty of Science, Professor Pushpa Chakrapani Madam, to address the staff and students.
Thank you, Kumar. Very good morning to all of you. Andariki Shubhodayam. I joined with Kumar in welcoming you all for this today's webinar, Biology to Biotechnology. At the outset, I am thankful to Professor Manohara Chari Garu, retired professor from Osmania University, who has a long association with Dr. B.R. Ambedkar Open University, and who is the today's expert and resource person to give this lecture in the webinar series. Sir, thank you so much for accepting our invitation and came to give a lecture in the webinar series, which is the first webinar series from the Department of Botany, Dr. B.R. Ambedkar Open University. I profusely thank our Vice Chancellor, Professor Sitaram Rao Garu, who is driving force and backbone for all the faculty members to conduct such webinars in the university. I'm also happy because the university is entering into the 40th year. We're going to have 40 years of celebrating 40 years of his foundation day of Dr. B.R. Ambedkar Open University. So on this occasion, all the faculty members are giving webinar series in all the faculties. So now the Faculty of Science has also initiated the webinar series. So thank you all for joining in the program. You all know that Dr. B.R. Ambedkar Open University is the first open university established in India in 1982. Since its inception, science programs have been started at a UG level, and now the PhD programs and also the research programs also started in Dr. B.R. Ambedkar Open University. So I'm happy to say that science programs are attracting and catching more of students in the society because of the quality education and quality product that is developed by the Faculty of Science, the department in the Faculty of Science. So once again, I thank Professor Manohara Chari Garu for taking time to give this webinar for our students. And it's my pleasure to invite Professor Manohara Chari Garu for this webinar. Thank you. Thank you, one and all.
Thank you, ma'am. Thank you.
It's my pleasure to introduce.
Yeah, now I request Kumar to introduce our resource person and expert of today's lecture. Kumar.
It is my pleasure to introduce today's speaker, dedicated Professor Manohar Achari sir. He has served Usmania University for 45 years as a Dean, Principal, Head, Chairman, Director in Usmania University, and as Vice Chancellor of Oriented University Indore. He has guided 50 students for PhD and published 640 research publications, besides 30 books, including 5 books by Springer. He has described 20 new genera and 82 new species. He did his postdoctoral at UK, US, and Germany. In 1999, he got DSc degree from Mysore University. He got 5 national awards, Best Teacher Award, Outstanding Scientific Award, and Young Scientist Award from Government of Telangana. He has served as President of BSI, IPS, IBS, and ISCA Plant Sciences. He is a Fellow of National Academy of Sciences India, National Academy of Agriculture Sciences, IPS, IBS, IMS, MSI, serving as an expert for UGC, DST, DBT, CSIR, ICRISAT, TERI, ICAR, UPSC, various universities, and others. His contributions are immense in biodiversity, taxonomy, conservation, biotechnology of microbes, fungi, and lichens. Presently, he is serving as a National Academy of Sciences India Senior Scientist. Now I request Professor Manohar Achari sir to give a lecture on biology to biodiversity, biology to biotechnology. Okay.
Can I start, madam?
Sir, yes, sir, you can start. There is a request from all from us. Students, if you have any doubts or any clarifications, please write it in the chat box. Or after the lecture is over, you can raise your hand and you can ask your doubts. Okay?
Yeah.
Professor Manohar Achari garu, you can start, sir.
Yeah. At the outset, I would like to thank the Vice Chancellor, Registrar, then Dean, Faculty of Science, and all other faculty members, and Mr. Kumar of Ambedkar Open University for having given me an opportunity to present this Biology to Biotechnology, a webinar seminar. It is interesting to know that one of the best universities, open universities, established in Telangana, and many of the eminent professors are now available in this particular university to teach and also to conduct various activities for the benefit of the students and disseminating the knowledge to the masses of Telangana and as well as other places. I thank one and all once again. Now I will be going for the topic biology to biotechnology, and this biology to biotechnology is one of the important aspects. Here I will deal all the aspects related to plant sciences so that the students will get the benefit out of this particular one. Yeah, biology is the science which deals about the life and its activities. It is referred as the biological science because it deals with scientific study of life forms. And in this particular life forms, and with reference to biology, we deal with the structure, function, growth, origin, evolution, and distribution of living things. Besides this, biology also deals with binomial nomenclature, classifies and describes the organisms, and how the species came into existence. All of you know that the evolutionary pattern and phylogeny, that is an important aspect to know how the plants and animals have originated on this particular globe, and the interaction between them, and also with environmental variables. And the 4 unifying principles are the foundation for the modern biology. and these are the cell theory, evolution, gene theory, and homeostasis. You can see on the left side, there are cell organelles. Cell is the fundamental unit of the life, and without cell, there is no life. And various cell organelles are present, which will be dealing with this particular organization and as well as functional activities of the animals and as well as plants and microbes and so on and so forth, including the human beings. So cell theory has become very important. And then how single cell, prokaryotic cell, that is where there is the nuclear membrane is absent, that is called prokaryote. From that particular prokaryote, how from the single cell, the multicellular structure like human beings and animals and plants and fungi I got evolved. So that deals about the phylogeny and the evolution. It also deals with, biology also deals with the gene, that is the material that is present in the case of the nucleus, that is the DNA, and that is chromosomes, and chromosomes have got the genes, and genes have got the DNA. So the DNA is the fundamental unit where all the life activities and hereditary characters are present in this particular structure. So gene theory has become very important in the years to come, and all these things form basic principles dealing with this particular biology. Now, every scientist or biologist, they felt that all these organisms which are there on this particular earth might have originated from a common gene pool. They might have got an ancestor gene pool from which the organisms might have got dispersed. And that is how they say that a basic gene pool might have been existing. And this universal common ancestor of the organisms might have originated or might have appeared about 3.5 billion years ago or more. So the life forms might have appeared in that particular, and they may be available in the form of fossils now. So biologists generally regard universally the genetic code as a definite evidence in favor of the theory of universal common descent. Actually, genetic code has something to do, and DNA structure has something to do with the physics also, madam. And a lot of physics work has been done on this particular, knowing the genetic code and as well as the DNA structure. So all these descendants like bacteria and archaea and eukaryotes. Archaea means the primary bacteria. The primary bacteria appeared, then the later on bacteria, and then eukaryotes. And all these organisms might have originated from that of a common gene pool. Now, gene theory is the one which deals about the biological form, how it looks and how it has to be, and how the plant has to look, animal has to look, and bacteria have to look, and then flowers, and as well as reproductive structures and all these things, how these structures have to be projected in the life form. So that is the biological form. And the function, what is the function of the gene, what is the function of the mitochondria, what is the function of chloroplast, and all these things are created from and passed on to the next generation by the genes. Therefore, gene theory forms the basis for the origin of life. And of course, Lamarckism and that is Darwinism, they are the two different theories. And Darwinism deals with the struggle for existence and as well as the adaptation of the life organisms to the different environments. And Lamarckism deals with different organisms that share the same basic machinery and that copies and transcribes DNA into proteins. So the whole biology is dependent upon the genome. It is the total complement of genes that is present in an organism or the cell. So the gene theory forms the basis for biology. This is the phylogenetic tree where I was telling that there must be a common gene pool, and the organisms that got classified according to Carl Woese into different groups. Bacteria, you can see, and this particular bacteria, you can see 7 or 8 groups are there: green filamentous bacteria, spirochetes, gram-positive bacteria, cyanobacteria, and then the thermoplagia and all these things. And archaea, they are also there like methanobacterium and halophiles, which are there in the saline habitats, and thermophilic organisms which tolerate high temperatures and so on and so forth. Eukaryotes means they are true organisms where you can see plants, animals, and as well as fungi, and as well as the other such kind of structures. You can see fungi and yeast and all these things, slime molds also they are present. So they are from that of a common ancestor, and still studies are going on to find out which kind of common ancestor has given rise to this. So in the early 20th century, the rediscovery of Mendel work led to the rapid development of genetics by Thomas and Morgan and his students. And after that, in 1930s, the combination of population genetics has come. And then now all these theories, starting from that of the Mendel's work, struggle for existence, and then the gene basis, and then Thomas and Morgan about the mutation and all those things, now they are considered as under Neo-Darwinian theory. So there are new disciplines that have rapidly developed, that is the genetics, and then genome, and that is called genome sequencing, and as well as the science of genetics and hereditary, and all that is dependent upon the model of the DNA that is created by Watson and Crick, for whom the Nobel Prize was awarded. So Watson and Crick have proposed the structure of the DNA, and this particular DNA formed the basis for the life. So the cracking of the genetic code is a major function, and that has led to the split between the organism and biology. Now we have got two kinds of biologies as on today, that is cellular biology and molecular biology. Cellular biology deals with the cell structure, functions, and evolution, and so on and so forth. Molecular biology deals with the genome complexity in a particular organism, human beings and other plants and animals, and as well as prokaryotes, and so on and so forth. So by the late 20th century, new field has become. Now there are three fields which are very important. One is genomics, where gene profile is available for many of the organisms. That is for the human being, or for the whatever it is, for the animal, or for the plant, or for the bacteria, and so on and so forth. Of course, for the human genomics, still it is under consideration and work is going on. And proteomics means that the protein profile, what are the proteins that are present in the case of the plants and animals, how they differ, and how to differentiate eukaryotes and prokaryotes. And then another third one is added, metabolomics. Metabolomics means every organism has got certain chemicals that is synthesized, and that the profile of that particular chemicals is considered as metabolomics. So this is the organism biologists using molecular techniques, and they evolved all these things, and the genomics, proteomics, and as well as metabolomics. So this is the recent trend that has developed in the case of biology. Now I'm coming to biodiversity. See, habitat is important, soil is important, water is important, forest is important, plant is important, animals are important, all these things are important. And they are present and they are living in a particular habitat. You can see in the case of forest, many of the eukaryotes and prokaryotes, and as well as you can see animals and plants and lichens, fungi, bacteria, and everything is there. So like that, forest itself is a multiple of organisms, and therefore it is known as biodiversity. I will give the definition. Biodiversity means variation among living organisms. Now you can see all the organisms are not the same. You can see, and all the fingers are not the same. All the faces are not the same. If you take the human beings into consideration, all the fingers are not the same. So biodiversity means the variation among living organisms from all the sources. Whether you take some organism from, for example, you take Eudogonium from that of the water. Eudogonium is different from that of the Cytonema. Cytonema is different from that of the Sargassum. Sargassum is different from that of the other organisms. And if you come to the fungi, Agaricus is different from the Penicillium. Penicillium is different from the Aspergillus. Aspergillus is different from the Rhizopus. Like that, if you take the plants, then Apocynaceae member is different from that of the Asclepiadaceae. Asclepiadaceae is different from that of the Papilianaceae. So all these things come under the variations among terrestrial, marine, and as well as aquatic ecosystems. That is from the water source, soil source, and as well as forest source and marine, sea habitat. So these are, and their complexes also. And besides that, it is a species to species, there is a difference. Within Oedogonium, there are 2 or 3 species. There is mild variation among these 3 species of Oedogonium. Even in the case of Rhizopus nigricans or Rhizopus other species, or Penicillium citrinum or Penicillium notatum and Penicillium other species also, if you see, there will be variation. So all this variation is because of the genetic complexity that is existing in this. So the differences between the species and between the genera and between the metabolites and between all these things is considered as biodiversity. That is, biodiversity is nothing but variation in all these aspects. Now coming to microbes. Microbes include microscopic organisms like cyanobacteria, that's a blue-green algae. They are not included in the algae now, they are under bacteria only. And because it is a prokaryote, and therefore they have separated that, now included under bacteria only. Viruses, and then actinomycetes. In viruses, you don't find any nuclear membrane, only DNA structure is present, either RNA or DNA. Actinomycetes, these are the filamentous bacteria. And bacteria is a prokaryote, and again, there is no nuclear membrane. Mycoplasma, both, of course, nuclear membrane is there, and then you will find all the structures. Some protozoans, and there is no cell membrane in the case of protozoans, chlamydia, archaebacteria, and then prions and viroids, and some fungi like yeast, they have included in the case of microbes. They have included yeast in the case of microbes because it is single cell structure, but it is a eukaryote. Even then, if it is eukaryote, they have included this in because it is single cell structure. Most of the microbes are single cell structures only. They are ultra microscopic. That is, you cannot see them with the naked eye and also with the ordinary microscope. You cannot see them under ordinary microscope. There is high powered microscope that is called ultra microscope. So the microbes are very important from the point of industry and so on and so forth. I will show you that. So microbial existence was not known till the invention of the microscope by Robert Hooke. Until and unless the microscope was made available, all these organisms were not known. So the discovery of the microscope in 17th century has made us possible to know what are the bacteria, what are the fungi, and what are the cells, organelles, and so on and so forth. So there are 2 kinds of microbes. One is autotroph, that is where the chlorophyll is present, and they are considered as autotroph. Algae is autotroph, plants are autotrophs, but some of the bacteria, they are not autotrophs. Fungi are not autotrophs, they are heterotrophs. That means they don't have chlorophyll. They cannot synthesize carbohydrate by themselves. They depend upon the one where the carbohydrate is available. That is the reason why fungi have become parasites and saprophytes. Even bacteria also have become parasites and saprophytes. Now, this is the biodiversity of the fungi. You can see on the left side in the upper surface, that is the entomogenous fungi. It is present on the housefly. You can see that there are a number of fungi which infect the insects. And if any insect causes the disease on the plant, and this fungus can control that particular disease. It is called entomogenous fungus. Entomo means insect, and genus means originated, entomogenous fungi. This is Beauveria bassiana, which controls the coffee berry borer disease. Now on the right side, you can see Saprolegnia, which belongs to Masticomycota. Earlier, it was included under Phycomycetes. And here you can see zoosporangial proliferation. That means within the one sporangium, many sporangia will be produced. These are called sporangia within sporangia. So multiple sporangia are produced, and it produces zoospores, typically biflagellate zoospores, and then they get released outside. So the zoospores are also, they show dimorphism. That means two types of, two generations of zoospores are produced in this particular one. So this is a peculiarity in the case of Saprolegnia. And you can see that within sporangium, another sporangia are produced, thousands of zoospores are produced. That means you know that the space is reserved, that is conserved, space is not wasted, and multiple zoospores are produced. ATP is also not wasted by this particular fungus. That is the peculiarity of this particular one. And then next comes the Pleospora as an ascomycete. You can see that slipper-shaped ascospores are there in this particular ascus. This is in the case of Pleospora. Then right side I have shown echinoderms. Echinoderms are used in the case of cosmetics. Anti-cancer drug is extracted from the echinoderms, and you can see that some of the fungi and the bacteria also they are parasitic on this particular one. Unfortunately, not much work has been done in India on this particular group. Now you can see another ascomycete on the left side. This is Neurospora. You can see 8 ascospores are there. And it is as per the Mendel ratio, the ascospores got distributed, and Mendel theory also has been proved using this particular one. Now, what I would like to say is this Neurospora is an ascomycete. It is produced in the case of Perithecium. And this there are, in the case of Aspergillus, you don't find a closer fruit body is there, and that is called Christothecium. In the case of Neurospora and Chaetomium, you will find only the what we call as Perithecium, where it is a flask-shaped structure within which that you will find at the base the systematically arranged asci, and you will find the ascospores. Here you can see 8 ascospores in each ascus. But the importance of this fungus is that 2,840 research papers have been produced working on this particular, by scientists working on this particular fungus. And you know, one gene, one enzyme theory has been proposed by the Tatum and Beadle, and who got a Nobel Prize for this. So like that, the lot of importance is attached to this particular fungus, and that is the reason why we have to study biology. Why biology is important? Because of this particular aspect. Then on the right side, you can see the Chaetomium. This is also an ascomycete. You can see setae, bristles are there on the fruit body. And this Chaetomium I am showing because not only it is Perithecium, but also I would like to say that it is important in industry. It elaborates and produces a lot of cellulase enzyme. And this cellulase enzyme is used in textile industry. After weaving, the cloth is very rough. To make it smooth, they apply this particular cellulase enzyme in industries. And in Surat, we have got diamond industry. And you see, to make the diamonds and other stones to shine from a distance, they apply this particular cellulase enzyme. That means cellulase enzyme is also useful in medicines. Cellulase enzyme is useful in the case of textile industry and also in garment industry. That is the reason why we should study organisms and why they have become important for us. That is the reason why we are going to deal with this particular biology. Then the next one, you can see on the left side, lower one, it is the Pythium. Pythium is the fungus which produces damping of disease. Phytophthora produces late blight of potato, and that is a serious disease which has become a famine in Ireland. Now here you can see Pythium, damping of disease produced by this, and you can see the zoospores, they get released into the vesicle. And into the vesicle, you can see the zoospores are released. And nobody has asked the Pythium to produce the zoospores into a vesicle, nor the Neurospora to produce ascospores in a sequence in an ascus, nor nobody has asked to have Chaetomium, that kind of bristles. See, this is the variation. Neurospora is different from that of Chaetomium. Chaetomium is different from that of Pythium. Like this, these are the nature itself has provided that the organism is can be differentiated by their structure and form. So only we are confusing ourselves. Otherwise, biodiversity is a very stable component, and structures are there as per the genetic makeup. The organism has got a structure like that only. Human beings have to be like this only. The monkey has to be like that only. Chimpanzee has to be like that only. The yeast has to be like that only. Amoeba should be like that only. So this is what the biodiversity means. This variation, and it is a genetically made up variation. Therefore, it is establishing there itself. Now on the right side, in the lower side, you can see bioluminescent ascospores are produced in a fungus called Ceratocystis. And this is the beauty of, see, the lot of physics is involved in the case of bioluminescent ascospore production. Now you can see, now I'm coming to Basidiomycota. Here you can see on the left side, Amanita muscaria, a fungus which is a mushroom, no doubt, but it is not edible. All colored mushrooms are not edible. You should keep your fingers crossed and one should not eat until and unless scientifically it is proved that it is edible and it is certified by scientists, and then it is multiplied by an industry and sold in the form of a mushroom like your button mushroom. So this is a poisonous mushroom. But the fungi are non-chlorophyllous because they don't have chlorophyll, and they are enforced to become parasites or saprophytes. This has enforced, absence has enforced them. Absence of chlorophyll has enforced them to become. And other one is they are eukaryotic also. Fungi are eukaryotic, and cell wall is made up of chitin, and chitin is the major component, and that is the reason why it has been created as a separate kingdom. I will come to that point later, but here this is the fungus which is poisonous, but nevertheless it forms mycorrhiza. You might have heard it that roots of the plants and as well as the fungus mycelium in combination, they go hand in hand, form a network, and that is called mycorrhiza. Myco means fungus, rhiza means root, association, symbiotic association. So wherever the mycorrhizal roots are there, then the fungal roots will absorb the nutrients and supply to the plant. And it is these mushrooms are present around the plants, around the trees. At the base of the trees, you will find many mushrooms in the forest growing. There is one fungus by name Amanita varna, a similar species, and that produces mycorrhizal mycelium up to 26 kilometers. And that 26 kilometers, that is, it runs the nutrients for up to 26 kilometers, and whatever the forest plants are there, they get the nutrients. Nobody is there in the forest to supply water, to supply phosphorus, to supply nitrogen and potassium, and so on and so forth. So it is the fungal mycelium, naturally existing mycelium, and 80% of plants, almost 80% of plants are having mycorrhiza. That is the symbiotic association of the roots with the fungus. Without that, there is no such kind of plant existing on the earth. So without mycorrhiza, there is nothing like that. So here you can see, but there is one importance attached to this amanita. Amanitin is the alkaloid which is extracted from this. It is used as an insecticide. All poisonous mushrooms, all medicinal mushrooms which are colored, they have got lot of chemicals. More than 2,000 chemicals are extracted from these, and they are used as a medicines. So whichever is not useful is now, whichever is not edible, they are producing a lot of chemicals for the human welfare, for the health, and therefore they have become very important. And you can see on the right side, there is an edible mushroom that is called gucci, and this grows under apple orchards. Underneath the apple plantations on the grass, you will find this particular one. And this particular, you see, this cannot be cultured, cannot be grown artificially. And like your Aspergillus penicillium, like a button mushroom, it cannot be grown. And so far, nobody could try, and nobody has tried to get this particular one into culture. But there are 7 species of this available in India. And every year, India is getting 4,000 crores by exporting this to other countries. They are getting 4,000 crores. See, where there is a will, there is a way. If you want to sell mushrooms, and if you want to cultivate the mushrooms, if you want to extract some chemical from a fungus or from a bacterium from the plant, you can do it. And you can start an industry also. Only thing is, we are thinking that wherever it is easy to get the things done, we are moving into that particular one. That is not good for the country like India, because the population is very high and we have to feed our population 150 crores, and it is going to be number 1. Then China will be number 2 by 2050. So our population is the major problem. Food security is very important. Medicines are very important, and climate change is also going to come. All these things should be taken into consideration. So this one is very much edible. And if you go down, there is one net is there. Net is laid down on this particular mushroom. This is called Dictyophora. As and when the insect sits on this particular one, it immediately dies. See, this is a beautiful fungus. And all these fungi are very beautiful, and we think that our faces are only beautiful, but fungi are beautiful, and flowers are beautiful, and bacteria underneath the microscope, they are much more beautiful than our faces. And now this is about, you can see Helminthosporium Drexlera, Carvularia, Aspergillus, and then Coltotrichum, you can see Acerulus. And why I'm showing this Coltotrichum? Because in Hyderabad, you can see cheetah moths. Cheetah moth is the banana, and banana has got more of, spots. If the spots are more, then the fruit is more sweety. It will be more sweet. So that is the beauty of this particular fungus. And it is no doubt it is parasitic on that plant, but because the fungus doesn't have chlorophyll. And then you can see the penicillium, from which the penicillin has been extracted by Fleming. Now, these are all the variety of fungi. You can see the diversity among the fungi. I want to show how they are different. And now here you can see broomstick-like structure fungus on the left side. This is a fungus which is broomstick-like structure. It's called Cinemata, and this comes under Deuteromycotina, and this fungi imperfecti. And there are so many other fungi like Carvularia also comes under that only, Alternaria and all that. Now, why I am showing this particular fungus? Because there is some importance attached to this particular one. It is not only a saprophyte, but also sometimes it is a weak parasite. And it is a weak parasite because it doesn't have chlorophyll. And then when L.V. Prasad Eye Institute has received some patients, 20 to 30 or 200 patients whose eyes have become red. And then they have asked us to come there, and we are seeing, and we have taken out the droplets from that of the infected patients, and we could find this particular fungus. So some of the fungi, they are also human pathogenic. They are present on the human skin, and as well as in the eyes, and as well as other organelles also. In the case of human, in the organs also you will find. If your habits are not good, the infection will be there on different organs in the body also. So skin diseases are very difficult to get removed. It is easy to get gonorrhea and syphilis very easily because of the bad habits, but to get a gold medal and silver medal in the examination is very difficult. What I want to say is that this particular fungus we have worked out, and we could find 0.02% of sodium chloride is given to them, and they are all right, and they have become all right. So another fungus is Trichuris spiralis. This is only to show variation and how the fungi have become very important in the human life. The mycorrhiza, as I told you, they are symbiotic, and these mycorrhiza, what they do is they associate with the roots, and they give all the nutrients, absorb the mineral nutrients from the soil and give it to the plant. And whereas the fungus gets the carbohydrate from the plant, and then it survives. So it is a mutualism. It is a kind of, so these are the types of mycorrhiza: ectomycorrhiza, arbuscular mycorrhiza, ectoendomycorrhiza, arbutide mycorrhiza, and all that. Right, 80% of plants, they have got naturally existing mycorrhiza, and without mycorrhiza, there is no plant that is existing on the earth. Now, this you can see the upper three left side. You can see they are all ectomycorrhizal fungi that you can see visually with your naked eye, associated with the forest plants. Specifically, if you go into the forest, you will find all around the plants some of these mycorrhizal fungus. They form network with the host plant, and some of them they cause diseases, no doubt. This is the other one is the lower one is the the other one is the arbuscule on the right side, and this is the vesicle. These are called arbuscular mycorrhizal fungi. That is WAM fungi. That here they are called, and they are also useful. Just I want to touch upon this, and they are very important in your syllabus. It is there. Then coming to the lichens. Lichens are very important from the point of environment. They are pollution indicators, and 25,000 lichens are reported from all over the world. And from India, 2,580 lichens are reported. It is, what is lichen? Lichen is a congruence. It is a combination of a fungus and as well as an alga. So mycobiont and photobiont, they go together and form symbiosis, and they symbiont, they become symbiont. So lichens are symbionts, and they are commonly found on bare rocks. You can see one I have shown you in the photograph. They are present here in the bare rock. On the rock it is there. On tree trunks they are there. On the old walls you will see. And when you move from that forest area towards Hyderabad, for example, from Vikarabad to towards Hyderabad, you can see that many of the rocks will be having these lichens. But when you reach Hyderabad, then it has stopped. So there is a lot of pollution in and around Hyderabad. Therefore, this lichen cannot be seen. But if you go towards Anantagiri Hills, Vikarabad, you will find such kind of lichens. So they are pollution indicators. And they may be crustose, foliose, and fruticose, and leaf-like or fruticose, like hanging like hanging structures. And then there are 700 chemicals which are there in the case of lichens, and that is the pigments are there, toxins are there, antibiotics are there, medicines are there, perfumes also prepared from this particular one, and then dyes. The earlier good olden days they used to use pH paper and litmus paper, and pH paper was used, and this is because of Rossella lichen, which is there available in India and elsewhere also. Now, algal diversity and biotechnology, and algae are abundantly found in water, humid soils, and rocks, leaves, subaerial parts of plants and other habitats, and they belong to Cyanophyta, Chlorophyta, Bacillariophyta, Phaeophyta, and Rhodophyta. Mostly Phaeophyta and Rhodophyta, they are macroscopic. You can see with their naked eye, like Sargassum, or you can see in the case of Polysiphonia and so on and so forth, and Laminaria, and all these things you can see with the naked eye. And Cyanophyta, blue-green algae, and Chlorophyta, like your Volvox, and then Chlamydomonas, and then Eudogonium. All these are the examples for this. And Bacillariophyta, that is diatoms, and they come under Bacillariophyta. And what is the importance of this algae? Algae can be used as a biofertilizer. Indian Agriculture Research Institute, they have taken Nostoc and Anabina, and they have grown under artificial conditions, and then produced a biofertilizer which can be used for the growth of many of the like rice and others, and farmers are using this particular one as a biofertilizer. And many of the seaweeds are used as a food and as a feed for the animals, as a food and feed for the animals and as human beings. Now you can see from that of spirulina tablets are available from in the market, and that is also known to all of you. On the right side, I have shown you an electron microscopic structure of a diatom, and you can see ceramic industry, and in the main that you can see, and in the defense also and space also. In the space, chlorella biscuits are prepared, and they will be eating, and it is not easily digestible. Therefore, for a longer time, they don't feel hungry. So in cosmetics also, many algae are used, and you can have beta carotene is from Durariella, Hematococcus, and all that. And nowadays, beta carotene is used in the case of cardiotherapy, when that is in heart diseases. They are using for the and also for productivity. Most of the lakes and reservoirs, in order to produce much of the productivity, if you want to increase the fish population, then the algae has to be used as a feed. So like that productivity of any kind of fishes and other prawns and other things, aquaculture, it is the algae which is used as a feed. And then algal blooms, some of them are Microcystis is a toxigenic alga, not all. And then pollution indicator, there are also pollution indicators. In polluted waters, you will find less of the algae, and also some of them also they are pollution indicators like cladophora and others. They will be used as a pollution indicators. And genetics, antibiotics, and bioremediation. Spoiling is by the human activity, whereas curing is done by the fungi, bacteria, algae, and so on and so forth. These are some of the Ophioglossum on the left side, and you can see Pinus cone on the right side, and in the below, you can see ephedra. Ephedrine is a chemical that is used. And then bryophyte, you can see on the right side, lower one. These are all primary land plants. After bacteria, lichens, and as well as fungi and other things, and algae, now the evolution has taken place, and now become the primary vascular plants are produced, and these are some of the primary vascular plants. You can see the cone and other things, and these are Ophioglossum, and that is a pteridophyte. And then you can see ephedrine is an important chemical. Yeah. And now plant fossils are also important. Fossils are the remnants of prehistoric plants and animals buried deep in the earth, in the layers of sedimentary rocks. Bacteria, algae, fungi, roots and leaves of flowers and fruits, seeds and spores are condensed, and they are now available in the form of sediments, and that is they are buried. And when you go for excavation, like in the archaeology, you will be getting some of the gods and goddesses figures. Like that also, you will be getting some of the dinosaurs and as well as some of the plants. So Ginkgo biloba is the living, it is the only species which is now available. Therefore, it is called living fossil. And the age of the earth is about 4.5 billion years. And in some form other, it occurs. And all these things are there: compressions, impressions, molds and casts, petrifactions. These are all fossil forms where you can see, and you can, if the in between the two rocks, if the leaf gets compressed, the leaf impression will be there on that particular one. And when you dig many of the deeper into the rocky area, when you find this particular one, then you will say that this fossil was present, this plant was present earlier in this particular area. That is the importance of plant fossils. So these are all Rhynia. You might have studied Rhynia, which has existed 415 million years ago in Scotland. And like that, different types of fossils were there. And the early life forms. This is Birbal Sahani. In Lucknow, there is Birbal Sahani Institute, and where a lot of work has been done on the fossil plants. Now, why fossil botany is important? Because we will be knowing the age of the earth, age of the plant, and age of the plant kingdom, age of the animal kingdom, age of the prokaryotes and as well as eukaryotes, and also where the oil is present, where the petroleum is present, and stratification of the area can be done, and so many other things are there. What was the flora and fauna, animals and plants that are existing some 1,000 years ago or 4,000 years ago or 1 billion years ago can be estimated based upon these fossil forms. So Birbal Sahni was the scientist who has established this particular institute. And plant biodiversity, of the more than 80,000 species are edible plants known to exist. And but human beings, that is farmers, are cultivating only 300. Of these 300, we are dependent upon 12, that is wheat and as well rice and some of the cereals and others. So pulses. So these are the 12 major plants on which we are surviving. And there are genetic modification and crop breeders, and they increase the use of plant species by using, that is, they increase the value of the plant species. Instead of 1 kilo of rice from one plant, they are now producing 2 or 3 kilos also, like that. As an example I'm giving, not exactly so, but what I want to say is high yielding varieties can be produced using biotechnology, and because they have discovered useful genes and which can be transferred from one plant to the other plant, and also from one organism to the other organism. And by transferring these genes into crops, now we have a lot of hybrids. In the case of rice, in the case of wheat, and we are self-sufficient with rice and wheat for another 20 years. And such number of 2,000 hybrids are available, and expand many wild relatives to modern crops. What they have done is, whatever the useful gene is there in the old plant, in the wild plant, which was existing in the forest, and that they have got it and transferred, and then they have become cultivars. All of them have become cultivars. So they have produced new hybrids, and that is on which we are dependent for our survival, for food security. So finally, the scientists all this has enabled the scientists to learn what important genes are actually contained in the millions of plant specimens housed in gene banks around the world. Now we have got gene bank where the genes of various plants, various bacteria, various fungi, and various animals are now available. And whenever it is required, they can transfer these genes into and produce a better variety. Now, these are some of the plants which I want to show. The plant kingdom is very big one. Geranium valencianum, that is one, this is present in Kumaon Hills, near Himalaya. And Artemisia, this is the plant which is immunotonic is prepared from this particular plant. And I'm showing only a few plants, not all. Erythrina variegata, and also you can see a common Himalayan subtropical plant, rubus. These are the fruits which are produced by the rubus in the forest, which are edible. And tribal people, they eat this, and a lot of medicinal properties are there in this particular plant. Now, these are some of the Cleanthus formosus and Lavateria. You can see on the right side, and then Scercoides campanulata, beautiful flowers are there. These are all from the Himalayan region. Now they have become endemic. This is a lichen which is available in the Himalayan region. And flame of the forest, Butea monosperma, during Holi, they prepare color. And then rhododendron is a big industry, especially in Karnataka. And they are exporting a lot of rhododendrons to other countries. And horticulture, floriculture, and as well as forest wealth. Everything is important, and plant wealth is important, and wild genes are there, and that we can use that particular, all these plants. And you can see Citrus rumpi and Rasula, the mushrooms are also there. Napoleana, beautiful flowers are there. And then I will come to, these are some of the orchids which we have collected from Eastern Ghats. And then you can see Dendrobium and Vanda, you can see Vanda also. The third one is, this one is Vanda, and that is pink colored flowers. And then like that, these flowers can stay for about 3 months. Some of the orchids flowers can stay in the flower vase for about 3 months also. There is one important chemical that is there, arginol, in the case of orchids. And that is importance, and a lot of uses are there in of the orchids. And now the orchid diversity, around 20,000 species are available in the whole world, and 1,200 genera, 177 representing 177 genera are there in India. 6% of world orchids are there in India. And I don't want to go into further details. In the whole world, we have got 1.5 lakhs of hybrids of orchids. And Thailand, with 400 species, they are earning a lot of money. And with 1,200 species in India, we are not getting much of the money. So that is how you can see. And then palynology, it deals about the pollen grains. You know the flower. And the ovary is there, petals are there, sepals are there, anthers are there, there is filaments are there, and then the anthers are there, stamens, and then in the anthers you will find the pollen grains, in the pollen sacs. These are the pollen grains. What is the use of pollen grain? Why we should study? Of course, everybody now they are not studying much of the morphology, but I can tell you there are a lot of uses of pollen grains. See, you collect all the pollen grains on a glycerin-coated slide, expose it to the air, and then collect the pollen grains and go to the microscope, and then see under the microscope, you will find different types of pollen grains. If you know the structure of the pollen grain, then depending upon that, you can say that these are the plants existing in this. Because every plant has got a separate type of pollen grain. That is the reason why you can say that these plants are existing in this particular area without going to that particular forest area. And then you can also trace the history of the plant species, how the plants have evolved. And then also radiocarbon dating, and then archaeology. It is useful in archaeology, biostratigraphy, petroleum exploration. If a type of, some types of pollen grains are present underneath the sea, then you can say that this petroleum deposit is there. And in oceanography, they are important. And then rural industries, their melisopelnology, that is honey preparation is done with the honeybees, and without insects and without pollen, there is no honey at all. Therefore, melisopelnology has become rural industry. Aeropelnology is important from the point of what we call allergy and diseases and other things. And systematic pelnology is important. And all these things are there for the applications of pelnology. Plant morphology is a neglected area. See, you should study root system, different types of root system, and then taproot system, adventitious root system, and then leaf structure, leaf margin, petiole, columnous leaf, petiole, leaf base, and so on and so forth, leaf margin, types of venations, and as well as axillary inflorescence, and also raceme and cymose inflorescence types. And all these things are necessary for the classification of the plants. And you have to, one should not neglect this particular area, and they are very important. And root apex and shoot apex anatomy is another important area. Now there is, coming to the biotechnology, there is no substance so important as DNA. It has provided a basis for evolution and also generated many millions of different organisms of plants, animals, bacteria, prokaryotes, and other things. And then they have occupied that are there on the earth since the first living organism which came into existence, as I told you, 3.4 billion years ago. And gene consisted of deoxyribonucleic acid, DNA, and the cells contain RNA in the cytoplasm and DNA in the nuclear material. And the nucleotides are adenine, thymine, guanine, cytosine. They are linked together by regular 5,3-phosphodiester bonds, and the double helix is held together by hydrogen bonds between the pairs. So this is Watson and Crick model, that is DNA. Now this is genetic code. See, among the ATGC, you can find that some of them, for example, AAA or UCA or GAC, like that, the combination will be there. So this is called the codon. And the codons are different types of codons. So there is two types. One is codon and nonsense codon also. So for each and every amino acid preparation, there is a code. So all these codons, they form amino acids, and all these amino acids, they form a chain, a polypeptide chain. And that polypeptide chain gives rise to a particular type of protein. And depending upon the protein profile, you can say establish the organism. So that is the reason why genetic code is important. It is the language of a particular individual that is spoken by the genetic code. So genetic code is the language of every individual. So you can see UAA, UAG, UGA. So like that. And here and there are nonsense codons also exist, because there must be a break for any kind of activity. Therefore, if the nonsense codon is there, then wherever necessary, you can introduce a new gene material and then produce new things. So this is very important. Now I'm coming to that genetics, that is first chromosome structure was discovered. Then later on DNA was discovered. Then later on they have studied what are the components of the DNA, and they have the DNA hybridization they have done. And then the sequence of the DNA, that is DNA has got the chromosome, the chromosome has got the DNA, and the DNA, that genes are which are made up of DNA in a sequence. That particular sequence of the genetic material has been done. And then later on, they have, for each and every organism, they have got molecular markers, and then heterochromatin and homochromatin, they are separated. And now genome sequence will give us a full picture of an organism, that is called science genomics and genomes. And epigenetics and population genetics, we know. So based on all these things, the cell is the basis, and then nucleus, and from the nucleus, the chromosome and gene, and then DNA. So all this. So this is only to show that DNA is the material, the hereditary material. You can see the bacteriophage. And when the bacteriophage infects the bacterium, E. coli, and then you can see the phage infects the bacterium, only DNA enters, and the empty bacteriophage will be outside, and then the parental phase DNA, they multiply, and then they come out of this particular one. So you can see entry of phase shell into this, and daughter phages assemble, and then afterwards you can find daughter phages coming out by bursting out the bacterium. This is only to show that DNA is the hereditary material, and in the new daughter phages, you can see bacteriophages, only DNA is present. So this is how the DNA has been confirmed as the hereditary material. Now I'm coming to biotechnology of various organisms. Now I've taken fungal biotechnology. Fungi are very important in bakery. Yeast. 50% of biotechnology is dependent upon the yeast. Yeast gives you alcohol. Yeast gives you bread preparation. No country can survive without bread and without alcohol. So a lot of income, and every country's income is based on this particular one. Cheese ripening in European countries is mostly because of the Penicillia species, and mushroom cultivation is a multi-billion-dollar industry. Say agar that is Pleurotus zogercazo, oyster mushroom, and then that button mushroom, Agaricus bisporus, all these things, they have become very important. So it is a multi-billion-dollar industry. In India, it could not take up, and I think India should go for this particular one. And it is why I'm saying this, because mushrooms are, they have got a lot of immune increasing properties, and it is anti-cancer, and it is anti-diabetic. A lot of fat is there, and protein is there, less of carbohydrate, and then out of 18 amino acids, each edible mushroom has got 8 amino acids. So it is good for health, and also is, in the long run, I think it is going to be medicine also in the years to come. So oriental food fermentation. You can ferment rice grains and as well as the wheat and all that. You inoculate the fungus. Then after a few, you add aroma, and then it will become a good fermented food. So you can use it as an alternate food in night time and other things. In European countries and USA, most of the people, they eat this fermented food only. And single cell protein, yeast is a single cell protein. Nira in the villages is single cell protein, because in that particular one, you will find the yeast only. Alkaloids, ergot alkaloids are the examples, and penicillin. Penicillin is the example for the antibiotic, griseofulvin. So 180 antibiotics are there. They are all from that of the fungi, bacteria, actinomyces. Streptomycin was the first antibiotic that was discovered by Waxman, and then from that of the actinomyces, streptomyces. And then later on, penicillin has become important. Ethanol, and then enzymes, uni-enzyme, and so on and so forth. Cellulase enzyme and lactase enzyme, and then lactase enzyme, which are used in industry. And then gibberellin, gibberellic acid, a growth promoter. Phytohormone is from that of the fungus Gibberella, and your seedless grapes are produced by spraying this particular one. Immunomodulator, that is organ transplantation by the doctors, is because of the cyclosporin. Cyclosporin is from a fungus, that is Talipoplandium, and cyclosporin, before doing any organ transplantation, they use this particular this as an antibiotic. Cyclosporin is used as an antibiotic, and after cleaning the organ with that particular one, they can go for. So it is organ transplantation revolution is because of cyclosporin, organic acid, polysaccharides, vitamins, and then paper industry, biological control, bioremediation. That is, wasteland soils can be converted into fertile soils by using biofertilizers from that of the fungi and bacteria. And ultra-coal is produced by this, and dye industry is important, and mycorrhiza is biofertilizer, and waste recycling. a lot of waste which is there in the forest, litter, decomposition, and whatever we throw in the domestic waste and industrial waste, all this is converted into soil and useful product by the fungi, bacteria, and actinomycetes. These are all some of the commercial enzymes: asparginase, amylase, catalase used in the medicine, beta-glucanase, glucoamylase, glucose oxidase, hemicellulase, lactase, and all this. So microbial, what are the, how the bacteria and actinomycetes are useful? They are useful mutation, understanding mutation, transformation, conjugation, transduction, and transinfection. And then plasmids are considered as a vector for genetic transfer, and also transposons and all these things are there. So how the bacteria are important? They are important in fermentation. Amino acid, for example, bacillus is used, E. coli is used, and vitamins, asparagine is a vitamin, and then antibiotics, penicillin and streptomyces, and enzymes are there from that of bacillus and yeast and trichoderma. Proteins are there, spirulina and yeast, and Baker's yeast is the common protein that you can get from that of the yeast. And food industry is dependent upon the fermentation, mainly is dependent upon these bacteria. But some of the bacteria, they produce toxins. One has to be very careful. And water quality, there is one E. coli test is there. And depending on that, by conducting this test, they will say that whether water can be drinkable or not. And dairy quality, and that is called as the quality of milk and other things is dependent upon this. And Streptococcus and Lactobacillus, they are probiotic organisms, Lactobacillus, and then probiotics is very important for the human health. And your ice cream industry is full of many of the pathogenic bacteria. Whether you want to eat, you can eat, but at least 2-3 pathogenic bacteria will be present in that. And whatever we send in the drainage and all that waste is converted into, it is degraded and then converted into useful form by bacteria, and waste disposal, that is Cellulomonas and Bacillus. So these are the important steps and important activities that is done by the bacteria. And this is biotechnology, the science of manipulating, altering biological systems. And this is animal, is a biotechnology project. By transferring the genes, they have done it. Unfortunately, because of tuberculosis, this has died. So this is a biotechnology product. So tools are very important in biotechnology. Techniques are very important. And we should try to gain a lot of things from that of by learning the things. Without techniques and without understanding the skills, having the skills, and without reading any of the material, you cannot do any experiment, genetical experiment. And the most important thing is by 2100, India is going to be #1. Even from 2050 onwards, India will be #1 in population. Now, biotechnology involves the use and manipulation of living organisms to get products useful for the mankind. That is, in fact, it is older as the human civilization. Earlier, they were also using this, and gene function is involved in this. What is that they will do is that the DNA they will take, and the DNA thread on one side is there, and another DNA is there, and a portion of one DNA, a little bit portion will be cut, and they will attach this particular DNA into the other one. So a new hybrid of DNA thread is produced. Like that, new organisms can be produced, or useful organisms will be cloned. So you can clone and produce this. So biotechnology is the one. So the tools and techniques used in the present day biotechnology, first you should synthesize the gene, isolate the gene, clone it, sequence, and transfer that particular gene. You have to sequence, because what are the components which are there in that has to be taken. So what is that? RAPD, PCR, DNA barcoding, gene sequencer, gene synthesize, microchips, and CRISPR technology. All these technologies are there, which you can take it for granted. And these are the techniques one should learn. And then single cell protein, India is suffering with malnutrition. So we have to increase the single cell protein content and control the malnutrition. So achievements in the field of agriculture, medicine, and industry have proved the usefulness of biotechnology, and it is now predicted that coming centuries would be dependent, mostly independent. But what is important here is biodiversity is important, organism is important, scientists are important, skills are important, clever genes are important. And see, without any clever genes, the organisms are producing so much. We have clever genes, but we are not able to do much work. The organisms are there to give a lot of products, and they are giving. Because of them, we are surviving. We think that we are great. No, we are not at all great. Our tongues are twisted, and we lie. And if for some sake of for our selfishness, we will also kill X or Y to get rich. And then every day, 100 and odd times of false, fake news will spread. And we are not disciplined. We don't have knowledge. And whereas organisms have got much more knowledge than us. They are very useful to the human society. And they are giving us food. They are giving us shelter. They are giving us medicine. They are useful in agriculture. They are giving us everything. Whatever life wants, they are giving us. But we are not, what we are doing is with our selfishness and with our jealousies, we are taking away the organism and selling it to somebody for the higher money. If that organism is lost, one medicine is lost. If that one medicine is lost, then many people will die. What is that we are doing? We are so selfish. I tell you, I think if at all there is any poisonous organism on the earth, it is the human being, not the others. They don't like each other. Whereas organisms, they like each other. They are very friendly with each other. Virus likes bacteria. Bacteria likes plants. Plants like the various products. See, that is the beauty of this organism. See, biodiversity is the resource material. So the technique of genetic engineering has been around ever since Stanley and Cohen discovered that applied recombinant DNA technologies to clone the first molecule. So this is how it is done. And a number of aspects of biotechnology can be dealt. Plant regeneration, cloning, variant selection, and mass propagation. By cutting the roots, you can grow a number of plants. By cutting for example, you take rhizome. If one rhizome is there, you can produce a number of plants from that. So meristem culture is there. Callus, cell culture is there. Anther culture is there. Tissue culture has become very important. And protoplast fusion is there. So all these technologies are available to multiply the plants, and animal cloning is also done. So this is one tissue culture experiment. In the case of Artemisia, we have done. We have grown that Artemisia plant in that flask, and then we have extracted the chemical artemisinin, and it is used as immunotonic to increase upon the immunity of the individuals. We have grown this particular one. So then we come to the metabolites. Number of metabolites are there. So plant tissue culture provides practically viable technology for commercially important natural products, bioactive chemical. From the leaf, there is separate chemical. From the petiole, there is a separate chemical. In the anthers, there is a separate chemical. In the root, there is a separate chemical. Vinca rosea, Catranthus roseus, and it is exported to other countries because it has got anti-cancer properties. Tulasi, that is Osminum sanctum, has got number of properties. Cup syrup is prepared from that of Tulasi. You know, Tulasi tea is sold in the market. See, these are certain things, common use is there available. So plant tissue culture provides all this material, and this is only to show that in the cell you have got plasmid, and this plasmid can be used. One gene can be transferred to the other plant with the help of this plasmid. So this is the proteomic genomics, and as well as protein profile and gene profile and metabolomics. See, I will come to the see this. How one has asked me, can we produce 2 TPH and 3D page figures. Then I said, yes, it is possible. Two-dimensional polyacrylamide gel electrophoresis, and also you can do. Somebody has asked me, in the school child has asked me, can I have Susmita's face? Yes, in the years to come, it is possible. Somebody has asked me, can we have Shah Rukh Khan eyes? Yes, it is possible. So it is going to be there. But only thing is, genetic engineering is very difficult experiment. What has happened in the case of Wuhan in China, where they were conducting experiment on viruses. And when that particular viruses they were conducting, then they could see that COVID virus, one COVID virus mutant has escaped from that. So that has become pandemic. Corona has spread to many, throughout the world, it has become a problem. Even today, it has become a problem. So to do genetic experiments, you need a lot of care. That is the reason why USA has asked the people only to conduct in the useful organisms, not in the case of the disease-causing organisms. These genetic engineering experiments have to be conducted only in the useful organisms with reference to crop plants, with reference to bacteria, with reference to fungi and other things where the genes are useful, not such kind of one experimentation. So this is not useful for you, but this is the neem. Neem has got 200 chemicals. It is grown in Asia, in India, in Middle East and Africa. Neem plantations are found in Australia and USA. I've seen with my own eyes in the case of Germany. And in New York, I've seen all along the roadside, they have got this plantation. Neem is the source of wide variety of biochemicals and widely agriculturally used azadirachtin as insecticide. And it is marketed naturally occurring insecticide. It has got great importance in sustainable and organic agriculture. See, you have forgotten the neem. And using this neem chemical, they are preparing your pesticides, insecticides, and as well as toothpaste and all that. Paste, they are preparing and selling it. Vajra, for example, you take Vicky. All these things, they have got neem inside that, and turmeric powder. So we have to enhance the human nutrition. The iron, zinc, iodine, vitamins remain a major cause of numerous human health problems in India, and the deficiency has to be solved. And for example, you take 127 million preschool children still suffer from vitamin A deficiency, leading to blindness and early death. So such kind of experiments have to be done. So golden rice, our ICR, Indian Council for Agricultural Research, they have synthesized it, they have produced the rice, golden rice, by transferring a gene which is responsible for beta-carotene synthesis. So we have got now rice of that particular quality. So now aromatic plants, WHO, World Health Organization, according to that, 80% of people who are there in the world, they chiefly rely, dependent upon traditional medicines. If you go to a tribal doctor, he will say that you take menthulu and eat them, your diabetic will come down. Like that, they will say different medicines. So for their health care, and major part of the traditional therapy involves the use of plant extracts. And you have got number of people are there, Ayurvedic doctors are there, tribal doctors are there who are curing number of diseases. So because of this, the whole world uses the plants and plant products. Many industries are established where they are getting the medicinal plants and extracting the chemical and synthesizing the tablet or the tonics, and they are preparing all this and selling to us. This is how, and India has got 1/3 of global biodiversity. We have got more medicinal plants, more fungi, more bacteria, more plants which are useful for us and other things. More good animals are there with us. So in the modern area, the use of medicinal plants, especially in the developing countries, decline. with the introduction of synthetic drugs as substitute for natural products. Earlier, we are using only plant products, but now synthetic chemicals are coming out. So we will see herbal drugs are there. And now these are some countries which use traditional medicines from the type of plant. For example, Europe, Japan, Asian countries, European countries, North America, and others. In India also, we use, and so many, these are the countries which are dependent upon, industries are dependent on this particular one. Now I'm coming to the medicinal plants. Adatoda xylonica, which is a common name is Vaza. Chemical that is present here is vaccine, and it is used for bronchitis. And if you want to reduce cholesterol, you go to Allium sativum. You have to use garlic every day. It will reduce cholesterol. And andrographis, there is one plant andrographis. It is hepatoprotective. And then anti-malarial. There is one Artemisia plant which is anti-malarial. So, and Bacopa monnieri tablets are available in market. They are known as memory enhancer, memory capsules they are called. So Centella asiatica for skin diseases, and Catharanthus roseus, as I told you, it is used as an anti-cancer agent. And haldi, curcuma longa, turmeric, is anti-inflammatory, antiseptic, and so many uses are there. More than 100 uses are there. So like that, we have got a number of plants which have got. Global market will be around 120 billion in the years to come. And global medicinal plants, we have got 30,000. So Indian herbal drug turnover per year is 4,000 crore. So we have to increase this particular one. And there is a necessity for prepare our own drugs for our population. So role of botanist, I don't want to go. Allelopathy. I want to tell you that there are two plants, small plants are there. And the third one, that in the center, there is one plant. So this plant is emitting some chemicals this side and that side. So it is used in the pest management. That means the central plant is able to control the insect which is coming to this plant and as well as the other plant. And it is also controlling itself. So there is stimulation and inhibitory interaction is there between the plants. So it is used for the control of pest management. So this is very important, and this is called allelopathy. This is called allelopathy. And you can use bioinformatics to classify the plants, animals, and as well as other things. And with the help of IICT, we have extracted the latex from Apocynaceae plants and plants of Asclepiadaceae. And we only based upon latex chemical component, we have classified the plants which are belonging to Asclepiadaceae and Apocynaceae. This is the one which we have got. And this is apomixis, formation of seed without meiosis and fertilization. So this is also a technology that is very important for us. And then plant breeding is very important to produce new and modern hybrids are from that of genetic improvement, and introduction of new genetic material has given us a lot of hybrids. And due to intervention of new technologies like transgenics and DNA, transgenics are also produced. The genetically modified plants are also produced. For example, your cotton, and you have got GMO. Modern genomics approach are efficient in gene discovery. So we have got a lot of modern technologies in our hand which can be used, and this is of course metabolomics and all that. And so as on today, we have got 210,000 chemicals, natural chemicals that are existing in the plants, animals, fungi, in plants, microbes, fungi, lichens, and other things. Aspergillus is now in 2019, this people from New Zealand, scientists from New Zealand, they have produced antioxidant from that of Aspergillus. It is a peptide. Lacidiploidine is another anti-cancer drug they have prepared from this particular one. So satellite remote sensing is very important in knowing the plant vegetation and as well as the forest also. What is the type of forest we have got, how to protect the forest and all that. And you know, as per the policy before independence, we have 33% of forest in India. area that is covered. But nowadays we have only 22%. 7% of forest has been lost after independence. This is very, very bad. And bioremediation can be defined as any process that using of microorganisms, fungi, green plants, and their enzymes to return the natural environment. If there is a waste soil, you apply a microorganism which is useful, like bacteria. Then the waste soil, the waste can be converted into fertile soil. So like that, bioremediation we have employed to attack soil contaminants, degradation of chlorinated hydrocarbons, and so on and so forth. So we have got a lot of things to do with this bioremediation process. And plant biotechnology development, the concept of modern biotechnology developed after science of genetics has progressed, and discovery of restriction endonuclease, ligase enzymes are revealed, and that has helped us to produce life-saving drugs, transgenic and beneficial microorganisms, and GMOs, and so on and so forth. And this is a transgenic plant that has been developed by us. And marker-assisted selection is an effective tool and comes without biosafety. Samba Masuri is susceptible to bacterial leaf blight. So you can produce number of disease-resistant varieties. And increase in population is a major problem. Therefore, we have to go for genetically modified organisms. And only we have to see that there is no toxicity in the product. And that is the reason why farmers are agitating for GMO brinjal. So 3 billion people are undernourished, 250 million children are at risk of vitamin A deficiency. So recombinant DNA, introduction of genes into rice, beta-carotene, we have produced golden yellow rice, scientists, and herbicide tolerance is possible because of vitamin A, and several other advantages are there. In future, we are going to have this carotene pigmented banana in the years to come because of genetic modification. So transgenic plants are produced and in the United States, and if we go to any other country, there are two, in the malls we have got two corners. One corner is genetically modified products, and other corner is traditional, produced by traditional methods. Whichever you want, you can purchase and eat. And that is how the people are using this particular one. So biosafety considerations are very necessary. Genetic engineering in the power technology has raised many ethical issues, biosafety issues, which need to be answered with regard to the deployment. There are some ethnical communities which don't want to eat genetically modified organisms because they think that their genes may get modified. So like that, there are some considerations. False notions are there, and we have to take care of this, and also biosafety considerations, that it should not become dangerous in the years to come. And technology transfer is important for us, and organic farming is now taking up a lot of emphasis laid on organic farming. It maintains soil health, vermiculture is there, biofertilizers are there, and biomonitoring, bioindicators there, animal health, vaccines are there, high quality of food and feed and fodder is supplied, post-harvest technology is there, plant health, we have got a lot of genetic resistance, and then biopesticides. Coastal ecosystem provide primary source of food security. We have to protect this. Now, abatement of pollution is very important, and mining and industrial areas have to be cultivated with the plants, and we have to protect soil erosion and enhance the beauty of the landscape and aesthetic value. And the Cine, that is the Cine writers, they sit in the garden and write. Therefore, beauty of the landscape is important. And without having the landscape, they cannot write anything. That is also there. So it also gives you a lot of mental peace when you sit in the garden. So all this is because of the plants and animals and all that. So this is Natarajan, one scientist is there who has produced a biofertilizer, that is cow urine, cow milk, cow curd, cow ghee, sugarcane juice, coconut water, banana ripe, and toddy. They are mixed together, and just like a tonic for us, it is a tonic for the plants. Many of the spices and other things are sprayed with this particular chemical, and they are produced in Kerala and Karnataka, and as well as in Tamil Nadu. Most of the farmers are using this particular one. Now this is pollution, and industry non-degradable are there, and I don't want to go into the details. Automobiles are very much there. And this is before independence. That was 93 million hectares of land was wasteland. But after independence, the wasteland has become 189 million hectares. That means after independence, we have increased the wasteland. Now that wasteland has to be converted and it has to become greenery by plantation and all that. So these are some of the scientists whom I would like to say that first one is Professor Swaminathan, is the founder of Green Revolution in India. Professor Ranjan, second one, is a plant physiologist. S.C. Satish Maheshwari, who has laid the foundation for tissue culture and morphogenesis in the plants from Delhi University. Professor C.V. Subramanian, late Professor C.V. Subramanian, he is from Madras University, and he has studied fungi. And K.C. Mehta, he is the fungi who worked on rust pathogen. For 22 years, he has worked on rust pathogen to control. Professor Sadasivan from Madras University, he has worked out on enzymes. Professor Mundkar is the present Indian Agriculture Research Institute is established by this particular man. And he is a late Professor H.Y. Mohan Ram on the upper side. He is an encyclopedia of botany. And Professor Chaturvedi from Andhra University is a good teacher. And then Professor Rangaswamy is a well-known, internationally well-known microbiologist from that of Coimbatore. He was the Vice Chancellor of Agriculture University. So nature composes some of our loveliest poems for the microscope and the telescope. So that is the importance of the microscope. And science is simply a common sense at its best. You have to understand the science. And one example I can give you is what is happening to the children is nowadays, they are going from Delhi to Gully with their Ramback shoes, and they are coming to the house and asking the mother or grandmother to give the food by sitting underneath a fan, which is on a heavy side in a chair. And you know, and the dust which is there in the shoes and the dust which is there in the fan, that falls because of the high speed, it falls into your plate which you are eating. There is an English saying that you are eating your own dust. So good habits are necessary. Actually, old people used to say that you leave your shoes outside and come into the house and remove your shirt and all that, wash your hands, wash your face, wash your feet, and then come and eat. And brush your teeth. Nowadays, people are allergic to even to brush their teeth also. That is, I don't know why these habits are increasing in the children and all that. Many people, present generation is not safe from that of the healthy, they don't have healthy habits, therefore they are not safe from the diseases. So science is simply common sense, it is best. Noble mission with a strong vision to build a great nation. This is the emphasis laid by late Dr. Abdul Kalam. So the revival of the village is possible only when it is no more exploited. We have to preserve villages, we have to preserve biodiversity. We have to concentrate on the village being self-contained. So what is important is biotechnology. Organisms are important, plants are important, fungi are important, bacteria are important, animals are important for biotechnology. Without studying them, without classifying them, without knowing their physiology, without knowing their morphology, without knowing their genetics, without knowing their ecology, nothing can be done. Therefore, this is the biology to biotechnology. And thank you very much for having given me an opportunity. Hello?
Sir.
I completed.
Yes, sir. Sir.
Yeah.
Sir, thank you very much.
Madam Chakrapani, madam, do you know that I am 76 years old?
Yes, yes, sir. నేను యూనివర్సిటీ జాయిన్ అయినప్పటి నుంచి మీరు యూనివర్సిటీకి వస్తూనే ఉన్నారు, మీ సర్వీసెస్ ఇస్తూ ఉన్నారు సార్. థ్యాంక్ యూ సో మచ్ సార్. థ్యాంక్ యూ వెరీ మచ్ ఫర్ యువర్ వండర్ఫుల్ లెక్చర్ సార్. ద టాపిక్ ఇట్ సెల్ఫ్ ఇస్ రియల్లీ బ్యూటిఫుల్ అండ్ వెరీ ఇంట్రెస్టింగ్. biology to biotechnology, and you have touched upon almost cell biology, molecular biology to biodiversity to biotechnology. You have explained beautifully with a lot of information. Hope all the staff members and all the students, those who are online, might have enjoyed the sir lecture. Sir, thank you very much once again, sir. And now I request if anybody has any doubts, I'll give you 5 to 10 minutes. You can interact with the sir. You can clarify your doubts. Thank you, sir.
Any doubts, please? Mansudan Reddy garu, namaskaram.
ఏమ్మా ఎవరికన్నా ఏమైనా అడగండి సార్, చాలా ఎక్స్పీరియన్స్ సార్, అంత మంచి సార్ మనకు వచ్చి మీకు ఇంత మంచి లెసన్ ఇచ్చారంటే మీకేమైనా డౌట్స్ లేవా?
They might be busy with the children and with wife cooking and with all that.
సార్ వీళ్ళంతా కూడా మన దగ్గర స్టూడెంట్స్ కూడా ఉన్నారు సార్ ఇప్పుడు ల్యాబ్ లో.
అవునా? వెరీ గుడ్.
సో స్టూడెంట్స్ ఆర్ ఆల్ ఆర్ వాచింగ్, లిజనింగ్ టు ద వెబినార్ అండ్ యువర్ లెక్చర్.
And they should ask questions, you know, that is more important.
Kumar, anything to say? Yeah. Now I request.
Sheikh Sahid, yes. He has raised his hand. Sheikh Sahid.
మాట్లాడండి. Yeah, ask your question. Sheikh Sahid.
Yeah, Sheikh. 3 participants raised their hand. Yes.
Sheikh Sahid, you can ask.
Yes. Those who have raised the hand, you ask the questions then.
Professor Madhusudan Reddy garu. Professor Madhusudan Reddy.
Madam, please unmute me. Unmute me. Unmute me. Please allow me. Yeah, madam. Now, can you audible, sir?
Sir, you're already unmute, sir.
Sir, madam, ఇప్పుడు వరకు మీరు అందరూ అన్మ్యూట్ చేయలేదు, మ్యూట్ చేసి ఉన్నాయి కాబట్టి క్వశ్చన్ అడగలేకపోయారు పిల్లలు.
చేయండి. ఓకే రైట్.
సార్ ఏంటంటే మనం ఇప్పుడు న్యాచురల్ రిసోర్సెస్ కి ఆల్టర్నేటివ్ బయో రిసోర్సెస్ అంటున్నాము.
అవునవును.
సో ఇప్పుడు ఈ న్యాచురల్ రిసోర్సెస్, మినరల్ రిసోర్సెస్ కి ఆల్టర్నేట్ బయో రిసోర్సెస్ అంటున్నాం.
అవును, ఎందుకంటే all around the minerals you do find some of these prokaryotes.
Yes, while extraction and while refining also there is a lot of pollution.
Lot of metabolites also they are coming out in that particular.
Yes, for that the answer we are giving is better move towards bioresources and biotechnology.
Yes. Bioresources is nothing but minerals also come under that only.
Yeah, sir. Number of
I tell you,
number of species so far we have not studied properly, sir. Still there are number of species.
Yeah, I tell you that I have been to Odisha. In Orissa, there is a copper mine. And in that copper mine, I could find that all around the copper particles of that mining area, I could find one fungus that is called mycorrhizal fungus. When we have cultured that particular fungus, it has become a biostimulant. It is a plant growth promoter. So like that, we have some of the, and it is also an indicator organism for that area.
సార్ ఇప్పుడు మనం ఈ బయోటెక్నాలజీ ద్వారా మనం ఇప్పుడు ఈ మెడిసిన్స్ మనం ఇప్పుడు ఎక్స్ట్రాక్ట్స్ వచ్చే ఏ ఉన్నాయి కదా సార్. ఈ ఇండస్ట్రీ ఇప్పుడు హైదరాబాద్ లో నేను పొల్యూషన్ కంట్రోల్ బోర్డ్ లో మెంబర్ గా ఉన్నప్పుడు దాదాపు ఒక 500 R&D ఇన్స్టిట్యూట్స్ కి పర్మిషన్లు ఇచ్చాం సార్ ఈ బయో ప్రొడక్ట్స్ బయో మెడిసిన్స్ చేయడానికి. కానీ వీళ్ళు అసలు అవుట్కమ్ ఏంటి, అసలు సొసైటీలో వీళ్ళు ఈ మల్టీనేషనల్ కంపెనీస్ తట్టుకొని బయటికి రాగలరా సార్? అదొక డౌట్ ఉంది మాకు.
మీకు డౌట్ ఎందుకు వచ్చిందంటే అది చెప్తాను మీకు. ఫస్ట్ ఆఫ్ ఆల్, in USA, Taxol was the multi-billion dollar biotechnology product, which is an anti-cancer product.
Okay, sir.
When it has come in USA, people were not using that as an anti-cancer agent. When it was produced, people were saying that no, it may mutate. There may be some sort of changes in the human mind and all that. So psychological problems were there. And another problem was that many companies were not coming forward to manufacture that particular product. Similarly, in India, we see purity of the compound is important. From you take any plant or microbe or the fungus, purity of the compound is important. Many people, from foreign people, they don't rely upon the compound that is prepared here. And that is the reason why they are not purchasing that particular one. So we have to establish purity and selectivity of the organism for a particular type of disease. And that is more important, because one plant may be having 1,000 chemicals. Out of 1,000 chemicals, you have to concentrate on a chemical which is highly potential and highly potent, and also which has got in a major quantity. And the purity is more important for any kind of product. So that is how the chemical companies are suffering from that. But I can tell you, tonics and other things, even other countries are also using from that. For example, you take Reddy Labs. Reddy Labs has become very prominent all over the world, and it is also using a number of plants. And we ourselves have identified the plants and given to them. We do have certain things, but at the present, some of the upcoming companies still they need a lot of experimentation at the field site, that is on the human beings and as well as on the animals and other things. That is possible, and I think in the years to come it is possible. And
సార్, ఇప్పుడు మనము వరల్డ్ లో ఈ బయోటెక్నాలజీ రీసెర్చ్ లో ఎక్కడ ఉంటాం సార్? టాప్ 1, 2, 3 లో?
Not 1, 2, 3. Don't ask whether 1, 2, 3. We are at 82 stage.
So thank you for your answers. And there is some Sudha T. He is raising her hand. Sudha, you can ask your question.
Ma'am, I am sir's student only. Sir, I am Sudha from Vanta College, sir.
Madam, tell me.
So just I'd like to wish him many more years of teaching, so that many younger faculty is going to gain the knowledge of his experience and wonderful lecture, sir.
Thank you, madam. Already I heard this lecture earlier also.
But every time I really wonder your energy, sir. Without a gap, 2.5 hours, you are able to teach, explain everything without missing any point.
Thank you very much, sir. All your
thank you very much, sir. Thank you very much.
Yeah, thank you.
Thank you, Sudha. Anybody? Yeah. Now Kumar.
Before that, madam, I would like to convey my congratulations to all of you and to the university. And I wish all the best and all the success to the university and to all of you in the years to come. And to the Vice Chancellor, to the Registrar, to Sudha Madam, and also Pushpa Madam, and Masudan Reddy, and also Kumar, and every one of you, to all the students, I wish a lot of progress for the university, and I'm very happy that it is becoming #1 in the whole of the country. Thank you very much.
Thank you, sir.
The dynamic leadership for all of you. Thank you.
Yeah, thank you, sir. Thank you so much. We always need your services. So we hope you will extend your services to our university whenever we are in need. Thank you very much, sir. Now I request Kumar to propose vote of thanks.
I feel honored and privileged having an opportunity to offer vote of thanks on this webinar, Biology to Biotechnology. First of all, I would like to thank dedicated Professor Murahari Chari sir to give a wonderful lecture. In his lecture, sir highlighted some of the crucial points such as cell definition and isolation of cell and isolation of prokaryote to eukaryote and diversity of microbes such as archaebacteria, virus, fungi, and algal members. And also highlighted biotechnology, recent trends such as PCR and genetic engineering and gene modifications and bioinformatics. They are all. And thank you, sir. Thank you so much for your valuable presence.
Thank you. Thank you very much.
I extended my heartfelt thanks to Vice Chancellor Dr. B.R. Ambedkar Open University, Professor Sitaram Rao sir, and a dedicated registrar, Lakshman Reddy sir, and Honorable Director Academic Sudharan Madam for giving an opportunity to conduct a webinar, and express my most sincere thanks to our Dean Faculty of Science and in charge head Department of Botany, Professor Pushpa Chakrapani Madam, for giving us support and guidance to make us successful this event. And I also wish to express my gratitude to all respected director and deans and faculty members. I must mention our deep sense of happiness for the computer branch for their immense support. I am grateful to thanks all the counselor teachers and dear students for their gracious presence. Once again, and thank you all.
ठीक है